community. He has worked closely with Lalji Singh Sir, and he's also an alumni of BHU. It's a great pleasure to have you amongst us, sir. We also have amongst us Dr. Rajesh Singh. He's the nephew of Lalji Singh Sir, and he's also the director of Lalji Singh Foundation. We are honored to have you, sir. It's an honor and privilege to stand before you today as we celebrate the remarkable legacy of Padma Shri Professor Lalji Singh, the pioneer of DNA fingerprinting technology in India, a scientist whose groundbreaking work laid the foundation for transformative contributions to human forensics, evolution and migration studies, sex determination mechanisms, and wildlife conservation. It has been a great privilege to have him as Vice Chancellor of BHU as well. Fondly revered as the father of Indian DNA fingerprinting, Professor Lalji Singh's unwavering dedication to science has left an indelible mark on the global scientific community. To perpetuate his visionary ideas and honor his contributions, AGNET instituted the prestigious Lalji Singh Memorial Lecture Award in 2020. This platform not only commemorates his legacy but also recognizes individuals whose pioneering research embodies the same spirit of innovation and impact. This year, we take immense pride in presenting this accolade to a scientist who has made extraordinary strides in the biological sciences and genomics, Dr. Rajesh Pandey. Dr. Pandey's illustrious journey began with a love for zoology, earning his graduate degree from University of Calcutta, followed by a master's from University of Delhi. His research blossomed during his doctoral studies at CSIR IGIB, where he explored the intricate regulatory role of non-coding RNAs in stress response. His passion for genomics then took him across the globe, from Johns Hopkins University in Maryland, USA, to MRC Harvard Institute in UK, enriching his understanding to genomics and disease mechanism. Currently, as principal scientist and group leader at CSIR IGIB, he leads the Integrated Genomics of Host Pathogen Research Initiative, focusing on unraveling the hierarchical regulation of host pathogen interactions, a field of paramount importance to global health and disease management. Ladies and gentlemen, today we have the distinct privilege of hearing from this illustrious scientist. Dr. Rajesh Pandey's illustrious career is not only a source of inspiration, but also a reflection of transformative power of science to address humanity's greatest challenges. It's now my honor to invite Dr. Rajesh Pandey to deliver the Lalji Singh Memorial Lecture. Let us welcome him with a warm round of applause.
Thank you so much. It's slightly emotional moment for me as well, because my initiation into science in 2004 September was through a project called Indian Genome Variation Consortium. And therein I started my research life as a project assistant, and it was my indeed privilege to meet Dr. Lalji Singh a couple of times during that time. And down the line 20 years, it almost like completes a circle where I'm feel a sense of gratitude as well to the initiation which I was initiated into. And it was through enriching interactions with him at that point of time which made me understand the strength of genomics, the power of asking questions which no other platform or stream of science has at this point of time. So with this sense of emotions, sense of gratitude, I will try to share some part of the work which we do in the lab, and that's why I have titled the talk as Genomics Teaching Us to Learn from Genomes. And when I gave this title, I thought what would be the essence of the talk. Then when the time came came nearer to reaching this beautiful campus, I realized that there is more than one coincidence, and I had clarity in terms of what to speak about. So I come from Institute of Genomics and Integrative Biology. The area of work is integrative genomics of host and pathogen. And the theme of the workshop is integrating archaeogenetics, forensics, and multi-omics for human health breakthroughs. So it's almost incumbent on me that I try to integrate different themes of work in the lab. And while I was listening to a lot of quality, interesting talks yesterday, some of the things which echoed was, for example, genomics is still a field of science which is cost intensive or on the higher side. How can we make use of that investment in terms of scientific return? How can we integrate different findings from the same data set? And what are the innovations which are possible? We just need to expand our horizon. We just need to open our mind that we don't know everything, but to be cognizant of the fact that there is so much to be learned from the same data set if we try to look into the data more closely without being caged by our own understanding that I know everything. I don't know everything. And same is true for me, same is true for my students, same is true for my lab, that we don't know everything. We are servants of science who are trying to understand what are there in the genomes which are trying to educate us in different ways. So through some of the findings from the work, we will try to understand that how the journey which started from sequencing first 125 SARS-CoV-2 genomes during the COVID-19 pandemic to doing something like this in the next 3 years, where we did sequence around 50,000 SARS-CoV-2 genomes in the lab, wherein our lab was the nodal lab for genomic surveillance for the whole of North India, with samples coming in at any time of the day for those 2.5 years, and how those understandings helped us to shape us as a lab, as well as made us responsible towards why we are doing research as well. And those work had many different milestones to it, but if I summarize, it included doing a lot of in-depth clinical surveillance, community surveillance, and those insights leading us to a lot many interesting ways of doing service to the society. And one of the pinnacle was that during the waves first by alpha variant as well as Omicron variant, we set up mobile sequencing at the Delhi airport, wherein individuals who came at Delhi airport and were RT-PCR positive were sequenced at the airport itself, and we gave reports within 24 to 30 hours in terms of whether they were alpha variant positive or Omicron variant positive at different stages during the. But then that's not the theme of the story today. What is the theme of the story? So that was the easier way of looking at pathogen and its different trajectories over a period of 3 years. But then we thought that's a comfortable domain to be in. So we thought of complicating us a little bit so that we don't have much of free time to do much of other things. So the theme which we choose for the lab was Engine Hope. And there's a small story which we deliberated for half a day in the lab, that what should be the theme of the lab. And when I was growing in research, I always used to not like much when a presentation used to start with scaring me that this many people, this million people, this 100 people will die because of disease ABC. Because my understanding is that we are anyways going to die, which I know today, when that's a different matter. So we thought, can we do some research which gives some hope to the public at large, to the patients at large, and whether that can give us a urge or a reason for us to work harder. So we decided to work on integrative genomics of host pathogen, with the idea that some of the work which we do in the lab will give some hope to the patients in the hospital through our different findings. So we work on different aspects, but please keep in mind that all the aspects which I have put in here are all coming from the same data set, which will get clearer as we go forward. So we work on the pathogen, of course that's the challenger. Then we work on the role of coding RNAs, we work on role of non-coding RNAs, Then we work on what's the role of co-infections, or in other words, we call it transcriptionally active microbes. So from RNA-seq data, in addition to the human reads, you also get non-human reads. So trying to understand what are those non-human reads trying to teach us.
Maybe you have a mobile phone inside the pocket Rajesh.
No, mobile I kept there. Huh? Stay. Is it? Yeah. That's right. We'll try it out. If it doesn't work, I'll shift to this. And then using what's there in terms of non-human reads, in terms of co-infections, then what's one gene has multiple isoforms, so looking into alternate isoforms, then looking into the perspective of if there are transcriptionally active microbes, what are whether they have potential to cause resistance, so looking at AMR, and then a different stream of work which is started 3 years. This is audible, I assume, right? Right? Awesome. Okay. Then we started that, well, this is looking at what's happening in terms of the bulk RNA-seq. So last 3 years, we have now invested heavily, not in terms of money only, but intellectually in terms of single cell transcriptomics, single cell antibody sequencing, T cell profiling, B cell profiling, to understand their roles in infectious disease. So this is a complicated story which we do, but then at the same time, they are all interconnected. How? We'll go to the journey. So this is one slide which you have to keep remembering for the rest of the talk. That all the 5 layers of findings in terms of pathogens, in terms of protein coding response, in terms of LNC RNAs, in terms of transcriptionally active microbes, and in terms of alternative transcripts, they all are 3-phased stories during the journey of SARS-CoV-2 or COVID-19 pandemic. So first one is the cohort 1, wherein we looked, this was when variants had not arrived, but then we had observation that different say infected by same pathogens but dissimilar disease severity. So we wanted to know what may be the cause of those dissimilar disease severity in terms of mild, moderate, severe, and different clinical outcomes in terms of recovery and mortality. Then we had intermediate phase where vaccination was introduced as a part of a strategy. We had vaccination breakthroughs. So we wanted to understand why a subset of individuals have vaccination breakthroughs compared to majority of us were protected against that. And the third part, we tried to look through the whole journey of COVID-19 and see, is there anything to learn from the things which happened before the variants arrived, during the peak of Delta variant, and during the peak of Omicron. So all the findings will be in that 3-phased work of cohort 1, which is looking into different disease severity, cohort 2, vaccination breakthrough and non-vaccinated infections, and third part, comparison across pre-variant of concern, Delta and Omicron. Then going forward, as I said, that theme of the lab has been integrative genomics. So what we have done as a lab which is more focused on how can we make use of the available money in a more cost-effective way without compromising the scientific findings therein. So what we have done a deep partnership with the hospitals in Delhi NCR and some hospitals beyond Delhi as well. And we get a lot of clinical samples with even a lot of clinical data. We use that clinical data to understand or to pick which samples we should invest deeper intellectually to understand different facets of these host pathogen interactions. So what we do once we pick up those samples, we do total RNA-seq because we are looking at an RNA virus which is SARS-CoV-2. we are also now looking into dengue, which is also an RNA virus. So your RNA-seq data gives you an understanding of the pathogen. Only thing which you need to adjust for is how do you set up your bioinformatics pipeline. Then once you have taken out the pathogen matching reads, in the subset of the follow-up data, you look at how you can have your bioinformatics pipeline to look for the protein coding genes. Then within those reads mapping to humans, you look for how you can look for non-coding genes as well. Then within that subset of data, you look into how one gene having multiple transcript isoforms can have a role in the disease trajectory, which if you remember, cohort 1, cohort 2, and cohort 3. Then within the same data, we looked into what may be the possible role of resident microbes or co-infection or transcriptionally active microbes, which is more comfortable for us to talk about. So this all 5 questions are being answered from the same RNA-seq data, albeit with different analytical pipelines. So in a way, if you trace a step back, if we are investing 25,000 for one RNA-seq, the cost of each of these studies is 5,000 per sample. So we are also doing cost reduction in a different way by asking multiple questions from the same data set. So where the journey started. So just taking a moment to say that where the journey started, and a lot of people whom I see in the audience today would be aware that, well, we wanted to see that what caused these waves. Because when we sequenced first set of samples in March, April 2020, we thought that we are so intelligent to have done such a good job of sequencing 125 samples and understood the ancestry and understood the origin of those viruses in India, not knowing that what is awaiting us down the line. So when the whole 2.5 years journey of COVID-19 was completed, in a way, we decided to look into the pathogen genome and try to find evidence that whether they hold some information for us, some teaching for us going forward. And believe me, that has been extremely useful when we are now working on dengue. So in this work, what we tried to look, we looked into a few thousand genomes across each of the time points or milestones during COVID, and we found that it's a matter of friendship. So during the non-variant days, which is the pre-alpha days, and a lot of people I know here are aware of linkage disequilibrium, so I will not invest time on that. But if you look at this red nose here, what they are essentially saying that during the pre-variant days, SARS-CoV-2 was only happy to travel with these variants together. So it had less flexibility. In the Delta period, it was still happy to travel with a set of variants, but it started to be slightly bargaining in terms of, okay, I can do away with some. And during the Omicron, it became very flexible. So if you trace it back and see the trajectory of the waves which happened, these were the ones where there was a shoot up in the infections and then it was sudden drop as well. So what did it teaches us that if the variants or if the mutations are going to be traveling together, you have a higher chance of not causing a much disaster compared to the ones when they become more flexible going forward. But that was not only the end of the story. What was also very interesting for us to was to note that during that Delta time, we heard a lot of murmurs and very impatient conversations around whether the drugs at that point of time were unuseful. And we thought that, well, in a retrospective understanding, does these genomes provide any information or understanding with respect to whether they can be useful? So we went back and looked at 2 sets of patients. People infected by Delta survived. People infected by Delta did not survive. People infected by Omicron survived. People infected by Omicron who didn't survive. And what was very, very surprising for us was that in the people who recovered, there were specific mutation background which facilitated remdesivir to bind with a much better affinity compared to the cases in patients which it did not. So these are Delta patients who recovered. These are Delta patients who did not recover. And this is a remdesivir binding site. Through molecular docking, we try to understand that how and we may explain what we see. And this was such beautifully captured through the mutations in the SARS-CoV-2 genome with 2 different outcomes, and exactly similar was the outcome when we looked at Omicron patients' background as well. That's why when you go back and revisit that sentence, that how genomes are teaching us that what we should learn going forward. What may be the implications of this? Can we do a dengue, for example, genomic surveillance for 5 years, backward and forward, and try to understand that which vaccine may be important? Because in our own opinion, we feel that we have too shallow data to come out with different vaccines and then feel sad at the end of the day that they don't have a high efficacy, because we have not worked hard to design and develop those strategies that how it may work. But this was an understanding which helped us that how genomic data has many such information. It's just your imagination which you need to unlock. Then we said, okay, we found pathogens to be doing what they do, but at the end of the day, we are doing some research which we want to benefit the patients, not to benefit or understand, or we are not in love with the pathogens per se. So we started to look into the patient's perspective, and few interesting insights which made us surprised as well. And one of those was this initial set of data where we looked into patients with mild, moderate, severe, and when we looked at their clinical data, we found that although they have different in terms of recovered and mortality and mild, moderate, severe, but when you look at their symptoms diversity, it is immense. So we thought, what may explain what we see? So we did a study on 125 patients with different disease severity of mild, moderate, severe, and mortality. And we did, as I said before, RNA-seq. I'm not going to go into detail, but what was interesting was that if we look into the profile, coding genes profile of patients who survived and who did not, very interesting, and to our surprise was that the patients who survived had very high expression of genes of mucin family of genes, and those were exactly downregulated in the patients who did not survive. So when we traced back that what explains what we are seeing, and these are the key findings from the other set of findings. So this way, if we translate into a public message, what we found was that in the patients who recovered, it had a more robust epithelial layer compared to the patients who did not survive, who had a leaky epithelial layer. Please do not interpret that in these cases virus did not enter. Virus did enter into both the cases because they were infected, and those were confirmed by many ways, including the whole genome sequencing of the virus itself. But what probably is messaging or the information here is that in these cases, our own immune system got with more time to mount an immune response compared to these ones where the entry was much easier and faster to happen. And then we traced back the other genes and proposed that how these findings help us to explain that why did we see differential disease severity. Then as we were going ahead with the work, we found that vaccination breakthroughs were happening. And then again, we looked into a subset of patients that why vaccination breakthrough is happening. And in those vaccination breakthrough individuals, you had less or mild symptoms compared to the unvaccinated individuals at that point of time, which is mid 2021 we are looking at. So we did take that approach. There are some major adjustments in terms of the analysis, but key findings I'm trying to take home message sort of thing. And one of the key findings was that we found very high expression for ribosomal genes in the patients, in the people who were vaccinated. And we were surprised that how come there are so many ribosomal genes coming up, because when we made RNA-seq libraries, we have done ribosomal depletion. So we are expecting that it will deplete those. But still we find that those to be highly expressed. We thought, okay, by any chance did we not do the experiment well? So we ruled out those chances. And then we realized that it's non-random in nature. How? And then we went back to literature and looked for whether there are any other evidences which are talking about ribosomal protein expression and SARS-CoV-2 or COVID. And we found that there were two articles at that point of time, but they had one line mentioned in the body of the manuscript. And then we said, okay, why is it so? Then we looked deeper into our data, and what we found was that there were two other instances when it was known for RNA virus, but in our case, we found that it is exclusively a higher level of ribosomal protein expression in the people who had vaccination breakthroughs. And then said why. So now if we go back and think that how a virus replicates itself, it's not a whole functional organism in itself. So it hijacks our own transcription machinery as well. And in the same individuals, we had high viral load as well. So when we correlated the both, we found that it does explain that why it will have high ribosomal protein expression, gene expression, and how virus would be using it for their own survival. But at the same time, it had down regulation of other key immune genes, which explained that it's a fine balance wherein in the vaccination breakthrough individuals, there is high expression of ribosomal proteins, which is probably being used by virus to replicate itself in higher number. But at the same time, vaccination is providing it adaptive response as well, which is helping the immune system to cope with the virus as well. So if you remember one of the line which I said that during Omicron, we had very high shooting wave of infections, and there was a certain dip as well. So it did not have a long tail during its infection period. So we said, okay, we are able to explain something which we saw on the ground. So this is summarizing what we saw in terms of the mechanism from based on the expression profiling of the genes and correlated with the clinical data. Then we come to the third phase of this work, which is trying to look at COVID-19 as a holistic way between pre-Omicron, vaccination, pre-variant of concern, vaccination breakthroughs, and the whole journey. And then we said, okay, what helps us to explain what we see from the host RNA behavior perspective? In this aspect, what we looked into that from the same RNA-seq data, we looked for the specific mutations in the SARS-CoV-2 genes and which of them may be correlated with or associated with the human genes. And what we found very interesting was that some of the key genes of the viruses which are important for its replication were having very strong binding effect. So we did a lot of molecular docking and other studies to show that those mutations were facilitating different dynamics in terms of the genes expressed during these three phases, and during delta, which we had a more severe impact on the ground, those interactions were impacted by the mutation background that had a role in the suboptimal immune response which we had during delta times. So if we now narrate the two part of the story, the findings from the SARS-CoV-2 genome and the expression of human genes here, we find that both are correlated in terms of mutations and their impact on these genes binding with this and effect on the virus's own replication as well as the immune profile of this. This is summarizing those findings in terms of graphical putting together that based on what we did, what we understood, what is that which we propose in terms of the findings. Then we shift to the third part of it. So we have looked into pathogens, we have looked into the protein coding genes. Now we are trying to look into non-coding genes. For matter of fact, it is important to note that when we looked at the initial RNA-seq profile of nasopharyngeal swab RNA, around 65 to 70% of the reads only map to humans. Please remember this number because normally when we do RNA-seq, we end up looking at the differentially expressed genes and not look at the whole bucket, that what is there in the bucket. We, being slightly cost conscious, we try to look that can we account for every bit of the reads which is there coming once we have made sure that the data quality and everything is taken care of. So of those 60% of the reads which map to humans, of those around 60% of that map to non-coding as well. So we thought why not to look at the non-coding as well and whether they can enrich our experience. So what we did was, again, if we look at, and this is the same cohort, please remember, the three cohorts which we are talking about. So what did we find? So we looked at one of the subsets in the data that is there any distinguishing LNC RNA between the patients who survived and didn't survive? And we found that indeed there were 3 LNC RNAs which were differentiating. And then we looked at the different severities in which they were expressed between mild, moderate, between severe, moderate, and we found that there were different set of LNC RNA. Then we did LNC RNA mRNA interactome study to look that if these LNC RNAs are going to bind with certain mRNAs, what is the expression of those mRNAs in our own data set. So these are some of the important mRNAs for which there are evidence of LNCRNA binding and which has a role in the disease context which we are talking about. So these LNCRNAs and these mRNAs data expression in a correlated functional way are coming from the same data set to help us understand much better that if we look at one dimension of it, probably we may not be able to explain or understand the complexity of the situation which we are looking at. And then we went forward and said that, well, are there a cluster of mRNAs which are behaving in a particular way? And we did find that across the different disease comparison groups, they are acting in a particular way. Then going forward, the second part of the study into vaccination breakthroughs and not unvaccinated infections, again using the same data set, we try to look into what may be happening. In this one aspect which we said, well, we don't want to be boring in terms of what worked for us in the phase 1. We employ the same strategy for phase 2 and phase 3. In that case, the life becomes very boring. We said, okay, let's look something else. So in this part of the work, we work deeply on what are the genomic features of those LNC RNAs which may help us to explain what we are seeing. One of the aspects which we have looked into, that these LNC RNAs had a higher density of repeat elements. What may be the functional role of those repeat elements? One of the ways is that probably they may be able to facilitate long range interactions, and one of the way to look into those was looking into topographically associated domains, and we did find that these interacting LNC RNAs had higher number of triplex forming potential. So they had certain repeat elements within these LNC RNAs which were facilitating higher number of interactions. So if it has to be a methodology through long range interaction, it has to be a three-way interactions. So there are ways to estimate this triplex forming potential, and we did find that in the interacting LNC RNAs there was they had the potential to do that. And then when we take it forward, that what are the reasons for it, we found that they are supported by the repeat element significant difference between those. So going forward, what are this? Then we picked up a specific gene example to explain the whole hypothesis that, well, if there is a particular LNC RNA which has a repeat element and it has the potential to facilitate long range interaction through topographically associated domain analysis, and within those we looked at transcription factors, and we did explain, picking up example that, well, this is how possibly it is acting. Now, whether that explains everything, again, as I said, that we are students trying to learn how many things we can learn. So in the third phase of it, we said, okay, we have certain LNC RNAs from the phase 1, phase 2, now come to the phase 3 and see whether there is more learnings to be made. In the third phase, this was more of a serendipitous discovery that some of the genes which you find here, this ADAR2 ApoBac family of genes, they were extremely high. They were non-randomly distributed between pre-variant of concern, during vaccination breakthrough, and Omicron. So we said, we got inquisitive that why this set of genes, which are pretty important genes for inducing RNA editing, why are they overexpressed? Then we went deeper into it and we found that these are actually again non-randomly distributed in terms of RNA editing within the LNC RNAs is much higher much higher in the Omicron and pre-variant of concern data compared to the Delta. So the one in the middle is Delta, this is pre-variant of concern, and this is Omicron, and the red one is your. So the take-home message is that we had higher expression of genes which facilitate RNA editing, and it was supported by higher levels of RNA editing in the same trajectory where the expression of those genes was. And it also explains going forward that why RNA editing is useful in cases where immune response has been comparatively better than what we observed during Delta. And then again, we picked up a gene to showcase that with an example that if there are RNA editing within those genes, how it has an effect through miRNA network, that those RNA editing impacted the binding of certain miRNAs, and how those miRNAs may be useful to to impact the expression of those mRNAs. And then when we went forward, we showed so we showed that expression of those mRNAs, so if you imagine the connections, it is like LNC RNAs, which had RNA editing, and those RNA editing impacting miRNA binding sites, those miRNA having an impact on the genes which they interact, and those followed the same pattern of expression, which explains why we saw different disease severity during these 3 different phases. So now we come to the 4th phase of the work, which is what is the role of resident microbes within us. And it is a known fact and understanding that when any infection happens to us, we already are in-house repository for many other microbes as well. So as in a social system, whether a person comes to us invited or uninvited, what may be our response? Are we always going to be very friendly, very welcoming, or we may have other engagements and we feel scared as a child, for example, that whether the chocolate or the sweets which we have may be shared by the guest now. So we thought, okay, we will look it from the microbial perspective and see what may be the response or what may be the functional role of the resident microbes within those individuals which got infected by SARS-CoV-2 but had different disease severity. Again, we went back and used our RNA-seq data where 30% of the reads were not mapping to humans. So we used those 30% of the reads and looked at where do they map to. And we found that of those 30%, almost 25% maps to the bacteria. And then, and please remember, 5% around map to SARS-CoV-2. So if you want to do a whole SWOT and the distribution of the 100% of the reads from a nasopharyngeal swab RNA-seq, around 65-70% map to humans, 30% non-humans, of those 25% bacteria, and 5% maps to virus. We didn't find much of fungi from the data, but that's the understanding from around 400 odd RNA-seq which we have done during this COVID time for selected group of patients. So we looked at, again, go back to the first cohort, which is looking at mild, moderate, severe mortality patients. And what is important to observe that in the severe and the mortality patients, there was a very differential abundance of reads mapping to the bacteria. Then we said, okay, what does it lead to? So because this was literally the first study at that point of time in later part of 2020, we were a bit stringent in our cutoffs and other things. And what we looked into that, okay, we look at the top 30 based on the number of sequencing reads supporting those findings and see what can be inferred from there. So these were some of the ones which were more or less present in most of the samples, but then downstream what you see is the differential abundance of those. So through a series of other interrelated analysis, we found specific bacteria to be differentially abundant between severity groups. Please remember these 2 names, Achromobacter psilos oxidans and Bacillus cereus, which were abundant in the patients who did not survive. When we found it, we were not experts of this to know at once that, okay, this is why it is. So we went back to the literature and tried to understand why it may be happening. We found other bacteria for other severity groups as well, but I will hold on or request you to hold on to that Achromobacter. So we went back to the literature and we found that this is because Achromobacter has a very high drug resistance potential. And then we went back to the clinical data and we found that these patients were more or less given the same class of antibiotics. So what may have possibly happened that in this subset of individuals, antibiotics did not have desired effect because they already had a high level of those bacteria which were resistant to those antibiotics. So that in a way explains what we saw. But then we also wanted to make sure, this is again going back to explain how can we explain the differential abundance and the functional role. But what was also important for us to look into that if we remove the identity of the bacterial species, can we look at the bacterial genes as a family and see whether there is differential abundance of certain functions of bacteria enriched in these samples. And what we found that top 3 were the ones in the patient severe and mortality. If you just keep focusing on this in different trajectory, you will find that ribosome is high in this. which explains that if there are higher abundance of those, it explains they will need this. Then bacterial motility, it means they were not cleared during their stay in the hospital, so they had lesser expression of bacterial motility proteins, and then bacterial chemotaxis also was lower in this. So even when we both put in the species identification or remove that and look only from the bacterial gene function perspective, both seems to come together to explain or help us understand that why did we find higher abundance of Achromobacter in the patients who didn't survive, as well as Bacillus cereus. And these findings were again validated in the second cohort, wherein we found that the patients who were unvaccinated and infected had a relatively severe disease, and again these were present in the higher abundance in those. So going forward, that's again the second part of the work which we looked at the core species and the variable species, and the findings going forward was that there was a common set of bacterial species in those which were looking into the functions were between commensals and pathogenic. But what is important to look into what happened in the vaccination breakthrough samples or the unique pool in those. In those unique pools, there were more of commensals in those compared to the opportunistic ones. Whereas in case of unvaccinated yet infected, there was more of opportunistic or pathogenic infections compared to the commensals. So the balance between the commensal and the opportunistic infection may also come together to explain or understand what we see in terms of the disease severity on the ground. So this is putting together the whole data in a perspective to explain how it may be impacting through the known functions of those bacteria in terms of the disease aspect we are looking into. So this is again for all our work, we try to give a message where I can explain it to my mom as well, that what we do in the lab. Otherwise she feels like I go at 8 and I come at 8, what do I do in the lab? So this is more for her to explain that this is very simple. We try to do this and we get this, not no any complicated cycles and other things to make you. And then when this part of the work was happening, my son was 5-year-old, so I had also to make him convinced that why I'm missing time to spend with him. So for all the work which we do, it's now almost a rule that we try to make a popular graphical abstract which can explain all the things which we do. This is again, if you remember, the species which we captured in the first part of the work in terms of disease severity, potentially markers, those were captured in this part of the work as well. Third part of this fourth chapter of the work was that if we look at pre-Omicron Delta and pre-variant of concern Delta and Omicron, do we explain the things in a holistic way? Because for the earlier two, we were more reacting to the situation at hand. But now when we are out of this, can we look at this holistically and explain and help us understand things for us to learn going forward? What we found as a summary of it, that in the Delta, during the Delta period, there was unusually high number of bacterial presence compared to the previous in Omicron. And please remember, these were all normalized for the similar number of sequencing reads, both overall as well as human, non-humans, same methodologies, same set of people, same set of sequencing platform, same set of analysis strategies. So there is no role of any of those. But what we found very interestingly, that this is what we observed in the data set. Going forward, when we look at a summary of those, what we find that in the Delta infections, we had more of opportunistic or pathogenic infections compared to the pre-variant of concern and the Omicron time. Okay. So, okay. Somehow the slide went off. So these findings we tabulated together and said that, well, this helps us, provides us some clarity in terms of what we saw on the ground during this whole journey of COVID-19 going forward. It doesn't like to move. Tired हो गया है. So now I will not hold you for long. We come to the fifth phase of this three-phased study to look into that if we know for long that there is one gene, multiple transcripts, how can that be useful and how can that be inferred from the same RNA-seq data. So one student decided to take that challenge, and one of the data set which we worked on was for a set of ICU patients admitted in the hospital. We collected samples at different time points and tried, and they were admitted with certain disease severity, and they were during their journey in the hospital they had change in their severity as well, that someone was infected with severe condition, ended up being mild and discharged, and some of them did not were infected mild but became severe, and some of them passed away as well. So we try to look at that dynamics, that from the same individual, if we look at the role of transcripts changing, and this idea did not come all of a sudden. When we were doing the other part of the work between coding, non-coding and stuff, we were finding that multiple transcripts were also there for a particular gene, but people who have experience in this will know that when you do differential expression analysis, lot of them ends up being normalized as well. So we said, okay, we will look at the individual roles as well. So in this work, we looked at a subset of patients with that background, and and this was the different disease trajectory. That same set patients admitted in the hospital had different disease trajectory while being admitted in the hospital. So one of the things we look into that whether there is any transcript diversity. We had looked into the gene diversity, and that's a separate work. We try to look into whether there is transcript diversity, and the answer was yes. There was transcript diversity when we compared two different time points of the same patient while being admitted in the hospital. And this is all the different transcripts which were captured from that data. Then we looked at if does RNA-seq data provide us a way to validate these different transcripts in terms of whether they are able to help us understand one of the ways of different transcript generation is alternative splicing. So we looked at the same RNA-seq data to find evidence for it, and one of the way of looking at it is using this leaf cutter tool to see whether from your RNA-seq data you are able to find evidence of different splicing sites within that data. And then we took this work forward, and then we looked into different severity between while they being admitted in the ICU, and we did find that there are different transcripts which explains that why they had different disease severity going forward. And then when we looked at specific genes going forward, we found that yes, there are differential set of genes enrichment in those as well. So when we looked at differential transcripts, we also then went back to a lot of cytokine data which was collected for those patients as well, and we found that those different transcripts underlying genes were also differentially distributed amongst different cell types as well, and that potentially may be playing a role in terms of different immune roles for those genes at different phases of the patient being admitted as well in the hospital. So if we combine those findings together, what do we find? So if you look at the inner circle of it, so it's differential gene expression, looking at transcript biotypes, looking at transcript diversity, pathway enrichment, looking at biological impact of it, immune cell abundance, and then looking at Bayesian network to understand these different aspects, which may explain that why a severe patient may have ended up mild or the reverse way as well. And that beautifully helped us to explain that what we are seeing here is something important to know going forward, because when we look at genes, we may miss the underlying messaging as well. And this is similar to if you imagine a difference between bulk RNA-seq and single cell RNA-seq. So going at a higher resolution to help us understand the disease in a different context. So now if I summarize all what I spoke for last 30-40 minutes is a single set of RNA-seq data to use it for different questions which are independent if we look from the outside, but at the same time they're interconnected as well, which helped us to understand different aspects of disease severity in case of SARS-CoV-2. And these findings have now helped us to go into single cell based work, again looking into different aspects from the 5 angles which we talked about: pathogen, coding RNA, non-coding RNA, single cell microbes, So what is the role of microbes present within the single cell and whether they help us to explain the disease severity, and then looking at those aspects. So, and we are now continuing work on dengue. So with this, I will be reaching to last 2 slides of it, some of the work which we are doing in terms of TCR dynamics to explain why do we see different disease severity for dengue infected patients, which is a work in progress. Why do we have for a group of patients having a lower respiratory tract infections, why some of them have pneumonia, some do not have? Can we explain that through the possible role of AMR? This is the work which is now done. So with that, I will pause here. There are a few set of slides which I will, seeing the clock, I won't be going forward. But this is the group lab work which has been supported by funders across the globe, including India and many parts of the world. And therein I will stop. Happy to interact, take questions, clarify.
Yes, Dr. Pandey, it's really wonderful work, and I'm very much impressed of summarization of your side. At a really good work. So, any questions or any information? Oh, yes. Yeah, really, I feel proud to chair this the Lalji Singh Memorial Award. Basically, it's an emotional moment for me also, as Dr. Pandey. And I'm a very long association with Lalji, and basically whatever I have done and whatever in the scientific community, because in India in the medical fraternity we do very little work. With the Lalji, I am really drawn to the scientific work, and I'm working on the discharge, a very new state in India. And their sickle cell anemia is a very high prevalence there. And with the Lalji's contribution, we do very wonderful work there. And that is, I ask a simple question to Lalji, sir. We don't have any systematic data in sickle cell anemia. At that time, there is a very few scientists working on sickle cell anemia in India. And when I work started, and he helped me like anything. From CCMB, he brought 4 scientists with his team, including Sangraj also there. He was collecting samples with Lalji, and we did about 20 lakh samples. It's a huge sample size, and we established the 10% frequency there. It is the his contribution. I today is a very proud moment for me remembering Lalji, and really I congratulate Dr. Pandey.
So if I could just add a minute, that what's the long-term impact of a vision, what you mentioned, that those findings have actually been instrumental for some CRISPR-based interventions work which is happening at IGIB for sickle cell anemia.
Yeah.
So those, establishment of facts that it is a matter of concern for us going forward. So those visions are like have a long-term impact as well.
Yes, as Dr. Pandey mentioned about the CRISPR work, we are associated. This is in the background. Lalji is the contributor for this, because for me, and because now we are in the team this CRISPR gene editing work with the IJ, Dr. Devu is my team, the director, the IJ. So with this team, we are working now in the gene editing. That is the notice for the gene editing in sickle cell anemia, CRISPR gene editing. So it is the only the largest vision was there, and we are now at that position. Thank you very much, and thank you, Dr. And inviting me and the team and chairing this wonderful session. And really, it's a proud moment for me. Thank you.
Yeah. Am I audible? No.
No, excuse me. Usually it's not a problem. If you have any doubt, you can interact with me.
So, during your CDIT cohort, so last time you did your study yesterday is in 2023, right?
Yes, excuse me. We usually don't call it any questions. You can interact later.
We will have tea time interaction. Don't worry. We will have tea time interaction.
Yeah. Yes.
Thank you, sir, for the fantastic lecture. It's my privilege to listen to chair for this session. It was very fantastic presented by the COVID your research.
थोड़ा आगे कर.
It's a very fantastic lecture which is given by you, sir. And we have learned several things. Last few years, we have the humanity was suffering from the COVID. And now we are doing this fantastic work. For the future, we are not going to this advanced kind of be. Thank you, sir.
Thank you for the flawless talk, sir. Your extensive piece of work, your vision, your research approach is, I'm sure, along with me, all the members of the audience will be inspired with. Now it's time to express our gratitude and honor Dr. Pandey for his exceptional contributions. I kindly invite Chairs Professor P.K. Patra, Dr. D.D. Singh, and President ADNET, Dr. Satish Kumar, to join us on stage for the felicitation. Dr. Pandey, we hope this token of our appreciation symbolizes not only our respect for your invaluable work, but also our gratitude for taking the time to share your experiences with us today. Ladies and gentlemen, please let's give him another warm round of applause.